Hey, this is Dan Heckman again. Now we're going to talk about some of the cells of the immune system after we just talked about the anatomy. So first of all, let's talk about innate versus adaptive immunity. Basically, innate immunity is the immediate, non-specific first line of defense of our body's reaction to potentially hostile threats. Adaptive immunity may arise after a prolonged innate response. This is a delayed specific response that remembers the pathogens and is triggered more rapidly if ever exposed to them again. So let's talk about some of the cells now. Here's a neutrophil. See those lobes? All right, so neutrophils tend to arrive first at a site of inflammation. What's this? It kind of looks like a stop sign. It's saying, stop inflammation. I'm releasing things that are degrading the pro-inflammatory cytokines. Okay, this is an eosinophil. They also have some other functions that we'll see later on. Adaptive immunity involves immune cells that are skilled in espionage and intelligence. So if you recall, you can think of your immune system essentially as the military for your body. So these are the James Bonds, are the adaptive immune cells. They take their time studying, carefully, reviewing the foreign pathogens, and scheming ways to combat an exactly perfect attack to those hostile antigens. Now, can you think of some examples? Here's a macrophage we see, which means big eater. Here's a dendritic cell, which we mentioned a little bit earlier. We also have T cells, right? Two major types. You know what they are? It's CD4 and CD8. So CD4 T cells are really like the positive feedback or the propaganda for the war against pathogens in your body. And so they release all kinds of cytokines that cheer on the immune system to keep up the good fight. Now, CD8, remember that it's kind of that silly mnemonic that I mentioned earlier. CD8, you ate it and it's dead, therefore. So they basically are silent killers and take out hijacked or infected cells of the body. Now, B cells, what do? Well, they turn into plasma cells, right? And they're involved in both types of immunity, actually, both innate and adaptive. So they'll synthesize some antibodies. IgM is the classic initial antibody that we think of for innate immunity. It's the first class of antibody that is produced by plasma cells. So IgG is evolved later on and is really associated with adaptive immunity. But there's several other classes that we'll talk about later. Okay, now let's discuss just how these cells, these immune cells, talk to one another and how they do that through presenting antigens to one another. And they do that through certain receptors on their surface, and there's two main types. They're MHC, which stands for Major Histocompatibility Complex, 1 and 2. So let's briefly discuss their functions. First, we'll start with MHC1 receptors. And they typically present antigen from intracellular pathogens, okay? So like this virus inside of the cell. They also present antigens from tumor cells as well. So how do they do this? How do they get a piece of this virus on their surface? So first, this virus, they can't put the whole thing up there, right? They need to just put a little fragment. So what organelle will chop up this virus? It would be the proteasome. Since this is going to end up on the cell surface, it then has to be imported into the rough endoplasmic reticulum. And then it can be loaded onto a protein called, I'll give you a hint, it's the title, MHC1. Okay? All right, here's kind of a tricky question. Can you remember what the specific subunit of this MHC complex that the viral fragment is loaded onto? It kind of is a chaperone for the viral fragment to the surface. I'll give you a hint. In patients who are on dialysis, this particular subunit is so large that the dialysis machines can't filter it out, and it can actually accumulate in the patient's bodies and lead to a form of amyloidosis. So it is the beta 2 subunit, all right? It's actually, believe it or not, that's actually asked on board exams. Probably because of the amyloidosis tie-in. All right, so the viral particle can be embedded in the cell surface where T cells and natural killer cells can take a look at it and destroy the entire infected cell that we'll talk about in a little bit. So first we'll talk about MHC2 receptors, all right? And on B cells, as well as other cells capable of consuming and degrading pathogens, such as macrophages. So what happens is a pathogen is phagocytosed or consumed by an inflammatory cell and stored in a phagosome. All right? Phage, think of food, think of eating. A lysosome, think of lysing or dicing or chopping up. A lysosome combines with this phagosome, all right, once the bacteria is in there, or the foreign pathogen, and it forms, well, they were really creative with this name, a phagolysosome. Okay, so now that we have this thing chopped up because the lysozymes combined with the storage compartment after it was eaten, we now have pieces of the pathogen ready to be transported to the surface. So the receptor antigen complex then travels to the surface and is ready to be presented to and stimulate other immune cells. All right, time for a flash quiz. What receptors mediate immune and non-immune cell interactions? Well, duh. MHC receptors. That was an easy one. Okay, moving on. All right, so it's important to know that MHC receptors that we just talked about are proteins. Thus, they are encoded by genes referred to as HLA genes. And the HLA stands for human leukocyte antigen. Just know that MHC1 is encoded for by the HLA-A, B, and C genes, and is expressed on almost all nucleated cells. MHC2 receptors are encoded by, do you know? HLA-DP, DQ, or DR. I don't know how they really named those, but they're special. They're expressed on all of, as we said before, the macrophages, the B cells, or the antigen-presenting cells. Now, can you think of a cell type that doesn't express MHC1? I asked this earlier. Red blood cells. Very good. Okay? They're non-nucleated, and hence they do not express the HLA genes. So sometimes the body may attack its own cells if the MHC1 and 2 receptors are too closely resembling foreign pathogens. Can you guess what examples of HLA genes are associated with certain diseases? For HLA1, let's talk about A3. Believe it or not, this is important to know for step 1, because when you're a doctor, you have to test for in the lab the HLA-specific types, and then you can know if a person is susceptible to some of these autoimmune conditions. So for A3, the answer is hemochromatosis. Okay? For B27, what do you think? B27. Well, you could think of the word PAIR. Okay? It's an acronym because it stands for psoriasis, ankylosing spondylitis, inflammatory bowel disease, and Reiter syndrome, which you can remember by the mnemonic PAIR. Okay, how about some MHC2 associated conditions? So this is a good one to know. The DQ2/DQ8 gene, what's that associated with? Celiac disease, all right? You just got to learn these, I'm sorry. Come up with your own mnemonic for them, or however you can associate them and retain them in your head. So HLA-DR4 is associated with rheumatoid arthritis. So just keep those in mind, scan through some of the others that are in your text, but yeah, come up with some silly mnemonics for me.